Welcome to Inside Cultivation. Today's a very special show. We are deep diving today in cannabis genetics. We are here with Kevin and Brendan McKernan from both Medicinal Genomics. They are both co-founders of Medicinal Genomics and CannMed, one of the most highly respected scientific cannabis conferences in the world, bar none. But before we get into genetics, let's talk about CannMed. It started in 2016 at Harvard. What the heck made you guys decide to start it? Because our industry definitely needed it.
Kevin, you want to give that one a shot?
So there's a bit of a circuitous path, but we actually started in the cannabis field in 2011, which has a bit of a history. We were sequencing a lot of tumor biopsies with these sequencers that we built. And we were noticing that they showed all this promise to maybe personalize cancer treatment according to the tumors that you have. But of course, the drugs they were picking from were like cisplatin and a lot of other highly toxic drugs to personalize. So we had a lot of people reach out to us saying, "What about all these non-toxic drugs that aren't approved?" And cannabis seems to help all of our symptoms from like a palliative standpoint. So that must be good, right? It's probably helpful for cancer. And that got me curious. So we sequenced the first cannabis genome, put it public. It's proven to be very hard genome to sequence, and by no means was the first few genomes put public. Are they anywhere near the quality of what's coming out today? I think we saw a great paper in Nature from Ryan Lynch yesterday, showcasing a nice pan genome of like 193 genomes that are going to be presented down at CannMed. And then it really picked up in 2016 when our father got cancer, and that really didn't occur for maybe halfway through that. But we also were seeing patients with epilepsy at the time benefit from this, and there was a CBD shortage. We had family members that were having AFib that seemed to benefit from CBD. So we realized that we got to bring all these people together and figure out how do we better monitor this stuff? How do we get better science wrapped around it? How do we get genetics involved so we can figure out who's going to best benefit from these things? And I'd say if you look over the course of that trajectory since 2016, we've learned a lot about the genetics of the plant and about who we can apply this to. There's now these papers out from Ethan Russo looking at cyclic vomiting. What they used to call cyclic vomiting syndrome, but we now know it as cannabinoid hyperemesis syndrome, is actually quite different than cyclic vomiting syndrome. And they have some genetic markers that predict which patients might be most susceptible to that.
Really? We've got advanced that quickly. How's the AI working with all that? Are you guys using AI?
Well, that was all actually pre-AI, so that's a
Really?
good point. I mean, this will probably get even better. But Ethan put together a study there. Granted, they had a hard time finding all the patients they needed because it is a rare disorder, but they noticed that there was mutations in the COMT gene, which is a gene that is known to be in the metabolism of cannabinoids. So these people are probably hyper-metabolizers, which is why they consume so much THC. And they noticed variants in the gene that capsaicin hits, the hot pepper spice. It's a trip receptor. And that kind of explains the hot shower phenomenon. These people all benefit from hot showers and capsaicin creams. It slows down the vomiting whenever they hit that. Yeah, and there was two other genes. One was a dopamine receptor gene, which is associated with people who have more addictive tendencies, which may explain why these people are consuming a tremendous amount. There's a fourth one I'm forgetting. I'll have to find the paper, but that was really, I think, the first time we've seen some really helpful genetics of the human population to understand how we can better, predict adverse events.
And by the way, you're well suited for this because one of the things is the Human Genome Project. You were one of the managers on that project. Would I like to talk about that? Because I remember you gave me the story when we're in Marco Island and we're sitting in the spot where 5 of you came up with the idea and then you made it reality.
Yeah, it was. In fact, Marco Island was where we go every year for human genome conferences. The AGBT conference be held down there. So the whole human genome was mapped out on that island. And
wow,
it was the whole goal there was to try to personalize medicine. Let's stop treating everyone like a herd and start treating people as individuals, which I think resonates quite nicely with the cannabis community.
Yeah.
So we're hoping we can do that with, better understanding the plant genetics and the human genetics.
Well, when you know human genetics and you know plant genetics, you put them both together. That's an unstoppable, unbeatable force. By the way, what makes CanMed so different than any other cannabis, like MJ Biz and Mary Jane Berlin are probably the two biggest ones, but when it comes to the science world, it's definitely CanMed. This is the brain trust of cannabis from all around the world, the top scientists meet. So what makes this unique and different than all the rest of the shows? I guess I kind of answered part of the question, but we'll hear from your perspective.
Well, it's go for it, Brennan.
Yeah, no, I think we're certainly proud that we attract, I think, the most data-driven minds in the industry. People that care more about the truth than what's trending. So I think you'll find at CanMed, it's really the who's who in the cannabis science world. You get everything from major MSOs to university researchers, clinicians, regulators, innovators, and even the clinicians that are treating patients. So it's a really interesting cross-section of the industry.
And cultivators. We have special cultivation panels, as we
do.
did last year and the last couple of years, that are very, very interesting. So you already answered the question, what kind of people should be attending? And it's, I guess, investors, innovators, PhDs, researchers, and cultivators, and anyone else who's interested in the science side of cannabis.
Absolutely. I think you'll see CanMed's not about kind of flashy boosts and hype. It's really about peer-reviewed science and clinical data that ultimately will drive policy and hopefully influence innovation in the field. So it's highly curated, and every session is handpicked to kind of push the field forward. It's really not just to fill a time slot or to get sponsorship dollars. It's really about building that ecosystem from bench scientists to physicians to method developers, and even the accreditation agencies are there. So when you bring that entire community together, it really has a tremendous impact on the field.
Yeah, let's talk about the networking opportunities, because you set up these very curated networking times for us to all get together and talk amongst ourselves.
Well, I think we try to kind of not only feed the brain, but feed the body. CanMed's a bit different than you'll see with traditional expos, if you will. We have curated workshops where you can have a deep dive. This year we have a very deep dive into genetics, everything from CRISPR technology and marker-assisted breeding, along with looking at the microbiome of the plant to see how you could influence yield and drive performance in the field, to the medical practicum, which is really a deep dive in the endocannabinoid system. And that's followed by curated talks that have been handpicked by a third-party advisory board. And then we integrate tremendous networking throughout the event. So every single meal is a sit-down meal, so you have a chance to really get to know your peers, which I think makes a very big difference in forging collaborations. And ultimately, our goal is to make a difference. So what happens at CanMed doesn't stay at CanMed. A little bit different from Vegas.
So
yeah, one aspect we try to emulate out of those days from the Human Genome Project was we noticed when you have an audience that's all staying at the same hotel, there's so much more interaction that goes on. Many of these other conferences you go to, everyone's dispersed throughout the city, staying at different places, and the moment the conference ends, everyone scatters. But all of the real innovation at those genome conferences happened like at the bar at night.
That's right, they do.
People were sharing notes and trying to brainstorm on how to do things differently, and that doesn't get captured in just sitting in a conference hall listening. Right? You need to take those ideas, run them outside, talk to 10 other people about them, and see how you can meld that information into your practice. So I think it's important you have it at a location that ensures and is very friendly to having that type of collaborative environment.
So speaking of locations, where is it this year?
Puerto Rico.
Puerto Rico.
Yes. Yeah.
No, it's exciting to kind of bring CanMed closer to the international scene. We wanted to reach out to the Caribbean and build a network in that region. And there's some really interesting players there doing some great things with the cannabis plant. So it's a great opportunity for us to bring sort of stateside to the surrounding outlying islands.
Okay. So that is going to be this June 17th through the 20th in, I believe, Rio Grande, is that right? Puerto Rico, at the Wyndham Resort.
Absolutely.
Yep. It's everyone in this industry, if you really want to know the truth, science reveals the truth, you should absolutely be going to CanMed and looking at that, because the connections that we made, the people that we've met, we've already done deals with a university. We talked to Texas A&M. We made a deal with Wisconsin University. And what I find is really fascinating is that the scientists a lot of them are they'll tell you if there's an NDA or IP that if you hit that, but they're pretty much an open book and really there to collaborate and help people and discuss things. And if you got a problem or a situation, I can guarantee you there's someone there who probably has worked on it or is working on it or knows about it. So let's move on from. Go ahead.
One last thought. I mean, I also, I'd like to give a shout out to you and your team because you've been a CanMed supporter since the beginning in trying to invest in the innovation of where the field was going to go. So you've been a very big part of the event and making this happen. So, we want to thank you for everything that Advanced Nutrients has done to support this effort.
Well, thank you for that. We appreciate it. But look, science reveals the truth. We've always been big on science. And when I heard about CanMed, it made 100% sense to me. Is like this industry needs a brain trust of where everyone can go and can talk. And whether you're a layman or whether you're a scientist, they'll talk to you in the language that you understand. I want to move on to genetics now. What do you guys think about this hemp loophole that has been created by the 2018 Farm Bill?
Oh,
that's
God,
that's a big discussion everywhere right now in our industry.
It is. So I think first off, we have to acknowledge when we started this, the hemp companies were the saviors. They were making CBD for all the epileptic kids, and they were really attentive to quality. And now there's a whole new, I think, hemp industry that's emerged that is probably not of the same ethos as those original players. And many of those players are still around. I mean, even for our father, when we were handling his cancer, we were getting these QC'd oils from Miriam's. I think they're called Miriam's Hope back then. Now I think they're Miriam's Hemp. Charlotte's Web, a variety of these really trustworthy providers making CBD. And they were very attentive to making sure the material was tested, because a lot of the physicians who are stepping out, putting their neck out to help these patients, they don't want to be administering random oils that haven't been QC'd, because that could backfire on them. There's pesticides, everything else. So that is an ethos you'll see at CanMed, is it does bring the physicians in, the patients in, and the growers in, so we can have these conversations to figure out what's the directive here? What's the best way we can grow high quality material so we don't have something like the FDA coming down and closing the whole industry. I mean, we're right now looking at Texas, where they're about to make a massive step backwards by banning all THC because the hemp industry flooded in there with all types of unqualified products, which is really unfortunate for the 1.5 million veterans that are in Texas right now. They're going to be stuck with the T-Cup program, which only has, I think, less than 1% THC allowable through the medical program, and hopefully they'll expand that to have more THC available through the medical program. But a lot of that was because Oklahoma was a neighboring state, and you're able to grow hemp and ship it in state, and it's under the guise of it being THCA, but you run those through testing laboratories and you find out it's THC. There's Delta 9 in there. It's above 0.3%, and it's going across state lines. But we did do a little study on this, which I can actually pull it up. It's a poster that we're gonna present down.
Okay, sure.
CannMed.
Yeah, absolutely.
And I think I have it ready for us right here. All right, so what we did, we went and took 30 samples, 34 technically, because 4 samples we ordered twice from the same vendor, and we ordered those from online providers who are selling things as THCA hemp. With they have an age check, but it's kind of a unverified age check. You just check a box on the website saying you're over 21, and they ship it. But we went and tested these for aspergillus and found 56% of them failed for aspergillus. So to put that in perspective, Michigan and Nevada's aspergillus fail rates are around 4% pre-shelf. This is post-shelf because we're getting these things off the shelf. So that means when you send material to those testing labs in the state, they test it for aspergillus. And they have 4% of the samples failing, those don't make it to the shelf. So presumably, if you did a shelf screen, like a secret shopper, it would be below 4%, maybe 1%. California, the numbers we've gotten out of California are less than 1% microbial failures in the legal market. All right, so this is really high from a microbial failure standpoint, and it suggests that the things that fail in the regulated state market just get repackaged and moved into the hemp market. We had Cambia and Analytica come in and run pesticide screens and cannabinoid screens. This chart you're seeing up here in the center is actually the THCA levels and the Delta 9 levels. And you can see all of them in the blue are close. They're all about 15% THCA. So this isn't a CBD plant. This is a THC synthase plant that just, they're harvesting it at the right time where there hasn't been a lot of decarb that's happened. I think only one sample was technically below 0.3% Delta 9, but they all had some Delta 9. Some of that is probably because the rules in the hemp field are that you get to test a couple weeks before harvest where the decarbing may not have occurred yet. Whereas if you dry this, you'll get more decarboxylation. And so on the shelf, it ends up quite different than that. We had 4 samples that were, in fact, those 4 samples were actually Delta 8 sprayed that CanMed picked up. And they know they were sprayed because when you make Delta 8 from CBD with the Lewis acid conversion, you get different isomers of Delta 8, and they were able to pick those up. So 4 of them didn't even have, were just CBD plants that got sprayed. They had some CBD in there, but it was Delta 8 on top of it. We then did whole genome sequencing on all of them. And the whole genome sequencing was interesting in that a lot of them were clonally related to one another, but under different names. So there's probably one core provider or a couple core providers shipping content of many of these online distros. And they end up getting renamed and sent out. The 4 samples we ordered twice and sequenced again 4 weeks later, none of the genetics matched the original time we got them. So they're just randomly throwing things in bags, regardless of the strain name. So what we did put together was like a chart here of the rapid growth of smoke shop licenses in many of the red states. You've got Georgia, Florida, and Texas here, all going exponential since the Farm Bill. So this stuff's getting sold into head shops or smoke shops under the guise of it being
and gas
hemp.
stations as well.
Gas stations as well, yeah. And there was a death in Georgia where a 24-year-old kid ate a 520 milligram Delta 8 gummy and went into a heart attack. Yeah. And then there was a fentanyl case up here. I think they're still waiting to confirm the fentanyl case, but there's one in Rhode Island. But some of these providers are also selling 7-hydroxymetragenine with these things. So they'll make 70% THC hash and throw in that kratom compound, which is technically an opiate, but it's meant to be an opiate that's less addictive than traditional opiates. So they're mixing 2 fields now. There's a lot of these players are in both the kratom space and in the hemp space.
What about all the synthetic cannabinoids that are out there? What are your thoughts? Which ones are the worst ones? And I can remember.
THCP I think frightens me the most because it's so potent. It's 33x more potent than Delta 9. And then, THC acetate's a concern if it's in the vape pens because that thing makes ketones when you heat it, and those are carcinogens. And that's getting sold into a lot of these pens because it's technically not on the DEA list. I'm not a lawyer, so don't quote me on that, but I understand the reason they can sell that in smoke shops, or they think they can sell that legally in smoke shops, is it's not explicitly Delta 9. The other ones, I don't know that I'm terribly afraid of those cannabinoids other than, are they pure, right? How are they synthesizing them? Are there side reactions there that we don't know about? I mean, there was a European paper that came out that showed novel cannabinoids that they were finding in some of the pens in Europe. I think they identified 2 in that paper that weren't seen before. So there is a little bit of a, Jesse Pinkman's kitchen going on here where they're just throwing whatever they can at the field to play cat and mouse with the regulators. Yeah. And I think what's most challenging about it is the hemp market now is banked, and the people who do the safety testing in the cannabis industry aren't.
That's right. Yeah.
So we know where that's going to lead, right? So they can do interstate commerce. They don't have the same age checks that you have in dispensaries, and not that I want to say that all that regulation is necessarily helpful in the cannabis market. Like, we were probably over-regulating the cannabis market, but there's absolutely none of it in the hemp market, and they have all these financial incentives from 280E to interstate commerce that is made them become. They're actually, from the market research we've looked at, it's now a larger market than the cannabis market.
It is, and it's in 31 states, and you see a lot of these big companies, the Tilrays, True Leaves, Curaleaf, that they've got into the beverage sector, the hemp beverage sector, and they've really blurred the lines with that. Now, those are big companies. I'm sure they're going to do things by the book, and they're going to get it right. But there's a lot of people out there that I'm sure there's synthetic cannabinoids in drinks and all kinds of other things going on out there. I can remember a time, personally, I thought they're all going to jail. I can remember walking through the Champ show, and I'm going, man, these guys are just asking for it. And I tried something and it had THCO, THCP, Delta 9, and HHC in it, and it was a 20 milligram. I said, well, I'll be careful with
had
this.
all of those in it.
I took yeah, it was it. So I just took a quarter of it at 5 milligrams, and I medicate at night to go to bed, so I'm pretty good tolerance. That just knocked me on my butt. It was incredible, and it was lasted for about, I don't know, 3-4 hours, maybe 5 hours, and I'm thinking, wow, this is definitely something different. And I don't know the efficacy and the safety behind all of that, because like what you said, they're just running wild out there. It's a wild, wild west. There's no regulation. There's no testing. So at the end of the day, like, what do regulators have to do to get a handle on this?
Well, there's a jurisdictional issue. In a lot of the states that have this going on, the government is somewhat cartelized in that the people managing hemp are in like the ag department, and the people measuring, the cannabis are in the cannabis department, and they're not speaking to one another. So we'll see. What we often see in response to this is the state testing regulators crack down harder on the labs for what there is certainly some lab shopping going on that's been discussed. They'll be discussed at CannMed. There's some concerns there. But the bigger lab shopping thing that's going on is people just not doing testing at all and running everything through the hemp channel. Like if it fails for any reason in the cannabis market, it probably makes its way there, and we can't get those two departments to talk. Like if you bring up the fact that, hey, you're tightening the belt on all these labs, they're all going out of business, and because there's a hemp market that doesn't test over here, can you get the hemp market to test? Like they'll say, it's not our department. Sorry, you just have to live with the fact that the market's moving in that direction.
Yeah.
So I don't know. Hopefully, we actually have a panel to discuss this very point down at CannMed, because it does require regulators. Like we got to talk to them. We got to keep the patients in mind, because there's also a faction of patients that want more testing, right? And not the entire rec market doesn't necessarily want to bear that burden. If they're not immunocompromised, why should all their stuff have to go through this, test it like it's uranium type of mentality? And so maybe the rec market should have different guidelines than the medical market. Some states do that. But I think all of that is kind of like moving deck chairs around on the Titanic if you don't address the hemp thing.
All right.
Because they're just, all the labs start suing each other, and it's a big fight over an ever vanishing market. And, in 5 years, there won't be a market there if they don't get their handle on the hemp market.
Sure.
And not that I would never be out there advocating to like, shut that down. Some of the hemp companies, really helped our father in making really clean CBD, but they did it responsibly. They put C of A's on things. We knew what we were getting. There's fair disclosure there. I think where it gets murky is when they're putting compounds on there and you don't know what it is. Like they tell you it's THCA, and then you run it through a test and find out it's actually a different compound they sprayed on it. And that's just not fair consumer confidence. So what I'm most worried about is that all of the sins of that market will get reflected onto the cannabis market that's trying to jump through all these hoops and do it right, and result in more restrictions on the cannabis market, like we're seeing maybe in Texas. And so we'll end up eating the cost of the loose activity in the other industry if we don't get our head around it.
Part of the problem also is the Analog Act. I don't know if you've ever read it, but Neil Gorsuch, that sits on the Supreme Court, when he was in the 10th District, he basically said the Analog Act is unconstitutional and unenforceable because of its vagueness. And he said actually under that Analog Act, chocolate would be considered to be an analog, the compounds in chocolate.
Oh, interesting.
So it's really, the waters are so muddied, and it only becomes illegal if you go to court and you're convicted by a judge or a jury that was an analog and you shouldn't have done that. Then that analog becomes illegal.
Okay.
I know.
Yeah.
It is. So let's move on to feminized breeding and loss of heterozygosity. So in feminized seeds, there's a lot of feminized seeds that are being used. And so what is the big concern about feminized seeds, and where is it going to go?
So that's been a debate since we've been in the industry for 10 years. More than that. God, we've been in the industry for 14, 15 years now. And I think this most recent paper will probably address it as well. That just came out. I haven't fully read the section on the par locus, but so everyone has asked, if we do feminized breeding, or what are we doing to these plants? We're the largest chromosome is the Y chromosome.
Okay.
It's over like 120 megabases in size. And obviously you do feminized breeding, that's no longer in the picture. So what does that mean long-term for the plant? So we finally just measured this because we got tired of like debating it online. And went about and grabbed 90 genomes out of Cannapedia. There's over 1,000 whole genome shotguns in Cannapedia people can download and repeat this on other ones.
That's yours, Cannapedia. And how many different, you've sequenced how many different strains?
We have 20, I think there's like 2,200 in there now, but about 1,000. Yeah. 1,000 of them were done a couple years ago. We switched to doing whole genome sequencing. When we first started, it was too expensive, so we were sequencing like 10 million bases in the plant that were near all the interesting genes.
Okay.
And then a few years ago, we switched to just do the whole thing. It's cheaper to do the whole thing, and with it, you get much more rich information. You also get the microbiome when you do whole genome sequencing, and so you can explore like the different microbiomes that exist in 1,000 different strains on Cannapedia.
What the heck does heterozygosity mean?
Well,
in layman terms for listeners.
Heterozygosity is a term for like how different the mother is from the father in the plant. Okay. So you have two genomes in any cannabis plant. You got the mother genome and it's diploid. You got the mother genome and the father genome. Now, there are some newer ways of breeding that make triploids and tetraploids. We'll leave those aside for now because those are another beast. But if you assume most of the plants out there are diploid, what you're doing is you're counting the number of variants that you see between the mother and the father. And that gives you a sense of like the diversity that's in the plant and how genetically inbred or outbred it is. So what you can do is take the genomes and measure that number, like measure how many SNPs do you see per genome or the heterozygosity rate. And is that different in males versus females? And is it different in one particular important region in the male? There's a 30 megabase section of the Y chromosome that's known to cross with the X. It has meiosis during it crosses over during male meiosis. So when the males line up and the gametes segregate, there's crossover events where the chromosomes can swap genes between X and Y. And some of this crossover event can also happen from Y to non-sex chromosomes. So Y can exchange small bits of DNA through structural variations and transposons into other autosomes. So it being the largest chromosome means we should pay attention to this, because if we selectively breed it out of the population by doing constant feminized breeding, we're not going to have the same gene pool on the Y that's mixing with all the other chromosomes. So this picture you're seeing on the upper left is a depiction of the part of the Y chromosome that crosses over with the X. That's known as the pseudo-autosomal region, or the par locus. And there are some interesting genes in there. One that's in there is in the gibberellin pathway, which is important for flowering. All right, so some of the flowering genetics are involved in this XY crossover. There's 110 pathogen response genes in this region that may play a role crossing over into the other chromosomes. So pathogen response could be something you breed out of the population if you're continually working with certain genetics. So the graph that you're seeing on the upper right there is just the tool we have on Cannapedia that figures out whether you're a male or female plant, and it just does that by looking at how much sequence do we see from the Y chromosome versus the X chromosome. That's what segregates the left and the right dot plots, and on the Y-axis there is the heterozygosity, and that tells you how much variation there is, and you can see it's a little bit higher for the samples in blue. That's because those are males. We see the same thing charted out in the bar chart down below of all 90 genomes. The males are on the left with blue text down the bottom, and there's pink text, which are the females. And we're looking at the heterozygosity genome-wide, which is in blue, and also the heterozygosity in the par locus, which is in orange. The par locus is this region that crosses over. You can see there's a lot more heterozygosity in the males in the par locus, also genome-wide. So they're more diverse genome-wide, and in that particular region that crosses over with X, they're particularly more diverse. So this is, I think, this kind of puts the nail in the coffin that, yes, there is more diversity if you are using some males, and there are people out there that are actually reversing males as a breeding pattern. Now, you don't get feminized or male seeds out of that. You still get a diversity of males and females, but they are doing reversals and getting some very interesting results and bizarre phenotypes. So males play a role. Now there isn't one solution to breeding. If you were trying to stabilize seeds, you want to drive the heterozygosity down. You want the seeds to become more and more like siblings that are more and more twin-like. So you're looking to kind of back cross them and drive the heterozygosity down so that when you make a batch of seeds, they all come out the same or relatively the same as siblings. So this wouldn't necessarily be your goal. If you're looking to find novel traits that don't exist in the current, THC dominant, Girl Scout cookies dominant world that we have, bringing in some males could make some sense. You might find, maybe people will find like hop latent viroid resistance this way, or they'll find other pathogen like septoria resistance by bringing in males and crossing them back into the population so that we can get some other traits reintroduced.
Yeah. What about genetic narrowing?
I'm sorry, genetic narrowing?
Yes. How's that going to affect things?
Well, so I'm not familiar with that exact term, but I'm assuming you're suggesting like the selection that might go on, like we have in the type 1 cannabis plants.
I'm talking about terpene diversity.
Yes, yeah. So it's a very good read. Ryan Lynch's recent paper, I thought, was really eloquent in this regard, in that they went into looking at the diversity in the terpene synthase genes versus the cannabinoid genes. The cannabinoid genes have fairly low diversity. Because we have been selecting for those, that's the dominant feature we have selected for over time is THC. And as a result, a certain set of genetics in that area has been hyper selected for, and we don't have a lot of diversity in those genes anymore. The terpene synthases, however, still remain fairly diverse inside of the gene, at least 193 genomes that they surveyed. And that resonates with what we found earlier with some work with Keith and Reggie when we did some terpene synthase sequencing and characterization in the past. Is that those genes, I don't think we have a selection going on in dispensaries for a particular terpene, right? It's rare they even display them. Some dispensaries will show off the terpene profiles, but probably only 10% of like super users understand. I want something that's got terpinolene in it, right? and half, sometimes the bud tenders don't even know if that's in the particular flower. So, I think until we start having those discussions at the storefront, there isn't going to be a consumer pull on the terpene synthase genes for some time, because there isn't a dominant one that consumers know to go buy, and therefore breeders are responding by breeding it. I think we might find this in another region of the genome soon, in that there is a lot of interest in the variants right now, right? There are people specifically looking for THCV and CBDV, and we're just beginning to understand the genes involved in that. The Phylos team presented on a region in the genome they found associated with variant production, I think at CannMed, maybe last year, the year before that. And I know Oregon CBD has done some work on this. But their most recent paper with Oregon CBD in Nature shows there's potentially a second region in the genome that's responsible for the propyl side carbon synthesis. And so maybe we'll start to see some selection for that over time as people start breeding for more and more of those types of plants. We'll get a sub-selection of plants that are really good at it, and only those genes will move forward, and the diversity will probably go down in that region as they begin to breed specifically for those.
What about genetic collapse, like in corn and bananas? Is cannabis going to face a genetic collapse?
So those have been, a lot of those have been GMO'd, and they have been bred to be monocultures and to be responsive to maybe glyphosate resistance, right? And so that, I think, has bred them into a little bit of a high production a corner, if you will. But the cannabis industry, it being so decentralized at the moment, where there isn't like an Anheuser-Busch of the cannabis field, everyone's got to grow in different states and use different strains, and there seems to be such a rapid strain turnover rate that we're not. I don't see that being an issue in cannabis. I think my only fear on that front is what we might be doing in the microbial testing space to push us maybe in the wrong direction. In that front, right? We have some states that are running tests that demand the plants be very low in total yeast and mold, and that's going to create a selection for only a subset of plants that can survive those tests. Zamir Punja put out really nice work this year. I think he's presenting this year at CanMed. He might be doing it remotely due to some.
Yeah, he's on one of our panels. Yeah.
Yeah, he's more of a panelist. Yeah, there's some. He's in Canada though. There's some restrictions getting him down there, so we're still sorting all that out. But
All right.
so one of the papers he published since then, he demonstrated that across the lines that he was growing in his facility, it looked like a genetic trait. Although he's not mapped where it is in the genome, there are plants, particular lines that consistently have 10 to 50-fold lower total yeast and mold counts over and over again, regardless if he changes the soil, the substrate, what have you. So they're genetically prone to have lower TYM counts. And we have those regulations in place that are basically wiping out the genetics that like to have a rich microbiome. That always worries us because we can't put a lot of faith on that test actually saving people. Like we understand the E. coli, Salmonella, Aspergillus. There's documented cases of people being hurt by those things, but we can't find literature on 10,000 CFUs versus 100,000 CFUs of total yeast and mold ever being a clinical consequence. It should be more of an indicator test than a pass-fail test, so that those genetics don't get totally wiped out. But that is an area that I'm worried about in the regulatory front, that we're pushing on that. It's something we lifted over from food, but I don't know that it necessarily has it's not clinically defensible. If Bros went and sued regulators for what is this arbitrary and capricious regulation you have here, according to Chevron doctrine, this shouldn't be around, they probably win because you can't find papers in the literature that support that test.
How can you how can you future-proof our industry for genetics? You guys are into genetics. What do you see coming, and how do we future-proof our genetics? What do we have to do as an industry?
Well, we're very interested in viruses and HLVD and pathogens, mainly because when we see numbers that got presented by I'm forgetting Jeremy's company's name, Darkheart. Is it Darkheart Genetics? When you see numbers like that of 40% yield loss, there is now evidence that there are some plants that are more tolerant to this than others. Some don't have yield loss with it, and so we've got to get better at identifying pathogens and early on, so we not suffer the yield losses, because when you have these yield losses, they just clean house and all types of other plants go in the bin, and we lose those precious genetics, probably forever. And so we don't want to see that happen, and so we're trying to build tools that better monitor these things, that look for not just HLVD but Fusarium. These things seem to run as dirty cousins, if you will. That whenever we see HLVD, there's usually Fusarium around. It's I don't know why. I don't know what we've seen evidence that in the literature that other viroids travel in fungal spores, and I don't think anyone's nailed that yet in cannabis with HLVD, but it's certainly the case for hop stunt viroid. They found it in I think it was Fusarium spores actually. They've even seen it travel in fungal mycelia. So we're still very early on the pathogen detection front, and I think that is an area that the better we can do at detecting those early and managing those, the better we can do saving some of the genetics, because the genetics seem to play hand in hand with this pathogen problem.
Okay, great. What gets you excited about any emerging technologies or research that you guys have come across?
Brent, you were about to step in there. I didn't mean to.
Well, no, I mean, I guess to your earlier question, Mike, my perspective on it is certainly genetic narrowing is certainly, it might be really good for brand consistency, but I think it could be dangerous for biological resilience. And I think that's something really important for the industry to continue to maintain the diversity of this plant. Because ultimately, I think it's not a nice to have, it's a must have to make sure that the next pathogen or environmental shift in this industry, in our climate, doesn't destroy a monoculture like we've seen with traditional crops. So I think maintaining that diversity and making sure that we don't breed ourselves into a corner over time just for commercial yield, I think that's a really important component that breeders need to consider over time.
Okay. What about IP protection and preserving breeder integrity? You guys are people can sequence, if they can bring you a plant and you guys can sequence it for them, and you have tests as well for Fusarium, hops latent viroid. I mean, you're a full service.
Yeah. Actually, there's this interesting story there that didn't get a whole lot of headline in that so the sequencing we do, there's an option for people to print a fingerprint of that data into Bitcoin. And that gives you a timestamp of when your plant was sequenced. And we've been doing that since probably, I don't know, 2015 or 16 for a variety of clients. And only very recently did it ever come to pass that some group got in an altercation about whose clone was whose and who stole it from who. They went to court, and the court upheld the timestamp on Bitcoin as being the evidence of ownership.
Right. On the blockchain.
It's basically, we don't always use Bitcoin. Some people, and more so now we do, because it's just become more of a dominant chain. But in the past, it wasn't so evident. We're sometimes putting them on Ethereum and Dash and Bitcoin Cash. But Bitcoin's transaction fees aren't extraordinary anymore. So it's still very easy to do this on Bitcoin. But that was the first time we actually had like a legal case that set the precedence that yes, these fingerprints on chain are proof of existence of cannabis genetics, and they can be used in court to arbitrate this type of dispute. So there's one functionality there. Now, you don't need to do sequencing to file a plant patent. You can talk to Dale Hunt about this. He's really well versed in this. It's a nice to have. It's not needed to have. But it does help if you get to a point where you're in dispute with someone over IP, because there's unequivocal timing of, you can do it the second you get the sequence in. You don't have to wait for the patent filing and all that stuff to give you timestamps. You can demonstrate that hey, at this date, the sequence was performed, and it probably costs $300 or $400 to do this and get it stamped, and then you've got yourself a record. In addition to getting that record, you get all the information about the plant, and the information for sequencing is becoming more and more valuable as all these pangenomes come out. We can make a hell of a lot more sense out of the cannabis genome today than we could 10 years ago. So it's one of these kind of like encyclopedias that just keeps growing on its own. You don't have to do any work. Just everyone in the world is working on the cannabis genome and annotating it. Your data gets more valuable over time as you can re-annotate it at a later date. And then you also get the microbiome information off this. So you can start to see, like, all right, what are the most common microbes that are in my plant? And is that something that, are they always there? Every time I do this, do I see the same ones? Does it change when I change my substrate, right? There's a bunch of information in there that we think's really important for obtaining yield and making sure your plants are healthy and not cultivating things that are going to get you dinged on the testing front. So we can see in the sequencing if there's aspergillus hanging around, if there's E. coli or salmonella, like those we know the frequency of those now across 1,000 different genomes, and we can actually predict to regulators like what are the things you're going to run into, what is the most common pathogen that's not tested for in the plant. One of them is Pseudomonas. That's in quite a bit of them. So the sequencing data has a lot of levels to it that I don't think everyone appreciates, and those levels are going to get more informative over time.
All right. So what does it cost to sequence a plant these days?
I think it's like $300 or $400 now, Kev. Is that right?
Yeah. We have chipped away at that cost over a decade, getting that down to be bare bones as possible for getting a whole genome shotgun. I think we're around 20 to 30x coverage. That's the redundancy at which we sequence. Okay. And it is they're now flipping them around. I think they usually load up runs with 20 to 40 of these at a time, and they flip them every week. So we had another 32 hemp samples run through just last week. So those will hopefully be updated for CanMed.
And I think that settlement Kevin was talking about, I think that grower received north of $400,000.
Wow, that's substantial.
Yeah, it was substantial.
Yeah.
So he felt pretty good. He felt pretty good about his sequencing efforts.
So they can bring a plant to you. You can sequence it, and then you can put it on the blockchain for them and fingerprint it, and it's theirs.
Yeah, and they have the choice in Canopita. You can keep your data private or public.
Okay.
I think Kevin, 95% of people put everything public.
No, it's not that. It's not that high. I think it's probably like 2/3. There's a host of people that are preferring to keep it private and analyze it themselves. We don't get like the studies that you're seeing here that I do, I only operate with the stuff that's public because I don't want to like accidentally give a presentation like this and disclose someone's data. So we have a flag in there that we don't do any analysis that goes public on private genomes. So you'll see the number you'll see on Cannapedia is the public number, which I think is north of 2,200 now. But I'm under the impression there's about that's only maybe 70 to 80%. We got to ask Steve. I don't know where the number. I haven't paid attention to the number recently, but it's not all of it.
Okay. If you're starting a cannabis company, what are some of the important genetic safeguards that should be put in place if you're going to be breeding your own or pheno hunting? What would you do if someone's just starting out?
I think that depends on like your goal. If I'm not on the sort of the business end of how to differentiate yourself in that growing market. And so some people want to go in and make seeds for everybody, and that's a different genetic goal than I want to make sure I have highest THC strain per se that never fails microbial testing. That's a different beast. And if you're outdoor hemp, it's a totally different beast. So it does require a bit of a discussion as to, what are you aiming for? Do you plan to have like 5 dominant strains and just keep using those year after year, or do you plan to rotate your menu every 6 months? And if you're in the rotation game, then you may have to pay a little bit more attention to yield, to things that affect yield or pathogens, because if you're rotating through different mothers and different clones, you're exposing yourself to new genetics every so often, and you bring a wrong one in, it can wipe out your whole operation. So the folks that are cycling through different strains have to have, I think, a bit more attention to QCing those strains before you put them in the operation. And that QC should look at not just top latent viroid, but look for Fusarium and Pythium and Aspergillus. Like those will come and bite you later if you reintroduce a clone that has those. And those are all things you can do at a really early stage. Like keep your new genetics isolated until you've completely tested them, and then graduate them into a new facility. If you're just working with the same like 10 cultivars and you're trying to keep them around with mothers for as long as possible, oftentimes we see growers who are doing that. They come to us and get them all sequenced, so they know, like, this is my stock. I'm going to have it for a long time. I want to have the genetics understood so that I have a record of this going forward, and I understand if I have. Are they genetically all the same? Like they want some diversity on their menu. We can help them sort that out on the informatics side.
Okay. Wow, that's a lot of information, a lot of technical stuff going on here. But that's why you got to go to CannMed, because you'll break it down. I mean, you use a lot of big words and terms, so to speak, and but when you get there, you'd be more than happy to use layman terms speaking to the folks out there. How do people get hold of Medicinal Genomics so they can get sequenced and they can find out? By the way, before I get to that, it's important that if you're going to put a new plant in, or you buy a mother from somebody, that you isolate it and you get it tested for diseases, because there's a large portion of people aren't aware that a lot of plants have diseases. What percentage of plants carry diseases right now that you see that most growers aren't aware of?
Well, we're probably the wrong people to ask in that.
Okay.
They tend to come to us when there's problems.
Okay.
So we would tell you an inflated number. When the house is on fire, the guy who puts out the fire would tell you fire's everywhere. But at least just looking at, so one thing we have on our website that I think is very helpful for all growers. We did this for regulators and to try to help the lab shopping consternations that are out there. Yasha Khan went out and FOIA'd 14 different states for testing data. We went and got Massachusetts and a couple others and to just try to confirm his work. And what's interesting in that testing data is it gives you a whole picture of sort of the microbial populations in time coming out of the different states. So there's a lab portal on our website. If you go to the Medicinal Genomics, just type in Medicinal Genomics lab data or testing portal, it will bring you up a page that will let you look at the E. coli, Salmonella failure rates, the Aspergillus failure rates, all the different tests they run, their failure rates across time in all the various states, and you can cross-compare them. And what's really interesting in that data is you can see seasonality in Aspergillus. Like, it tends to spike even in different states, probably because everything's done indoors, but in Nevada and Michigan, it tends to spike around like August, September timeframe.
Okay.
And it goes from like them having 4% fail rates up to like 10% fail rates in those months. And it repeats itself the next year. All right. So there is some seasonality to this, and there's some data that people can use to perhaps, prepare for this or better predict these things. But that is one aspect we try to bring to the understanding of the genetics of people's plants, is we try to couple it with all this testing databases we have. So we can say, look, we know the frequency of septoria in cannabis. We know the frequency of Fusarium. In fact, we've found, in collaboration with Zamir and a couple other people, we have found novel Fusarium species in people's plants. They're now submitting them to NCBI and publishing papers. So there used to be about 4 Fusarium species people would talk about in cannabis. We now have the list out to 10.
10?
And tests
I knew it
that
was 7,
target
and
all
now
10.
it's up to 10. Wow.
Yeah, it's up to 10. And so you have to refactor your qPCR tools every time you find a new one. You got to make sure, does our test hit it or does it miss it? And we got to disclose that if we can't find it, and then redesign it so that we hit it. But there is value in the testing data that's out there, and as to how it can inform growers that are even for some of the organisms they aren't being tested for. Like Fusarium, no one tests for a regulatory standpoint. Are the growers come in and ask for it all the time because they know they got it, and it's a major yield problem. But no regulators asking for that to be tested for. But we have that sequence information in these 1,000 genomes. We can go through and chart out the different Fusarium species that are present there, and we know the frequency and the sort of reads per million that we find it at, and we can segregate that based on state and jurisdiction. So there's a tremendous amount of data in those whole genome shotguns that can help guide how to breed so that you don't get tangled up in either a yield problem or in a Aspergillus, E. coli, Salmonella problem.
Mm-hmm. So which states do you find that the most diseases are coming out of right now?
I have to go and check on the reads per million for these things in different jurisdictions. It's a good question. If you just look at the testing data alone, Massachusetts actually has being a wetter climate out here, we have an elevated failure rate for yeast and mold.
Okay.
Now, California no longer uses that test, which is probably the right call, and as a result, their failure rates are below 1% for micro. You go into Nevada, Michigan, the Aspergillus testing rates are closer to like 4%. You go to Alaska, for some reason, they're really low up there. Hawaii is a very low rate, but there could be artifacts in Hawaii. There's only one lab in Hawaii, and so there could be a method artifact there at the lab that just undercounts. So some of that has to be carefully interpreted based on what methods people are using. Some people are using plating, some are using microarrays, some are using PCR. And if you don't dissect it, you can draw some kind of hairy conclusions. But I can't say I can place blame on one state being worse than the other right now at a grow level.
It sounds like our industry needs more standardization.
Oh yeah. I mean, I'm glad you hit on that, because we're running tests that have no standards. Like, that's the insanity of the cannabis industry. Like, there are no real standards for the total tests, right? There is no such thing as a total yeast and mold standard. Yeah. They tried to make one for a while that had 2 organisms in it, just 2. But you can imagine that's not a very good standard when you're supposed to be testing for like 10,000 to 100,000 different organisms. Yeah. So that's bad. We have standards for the species-specific tests. E. coli, Salmonella, Aspergillus, the ones that we know what we're targeting, those things are solid tests. When you get into the total test, there aren't standards, and it's all a little bit of tarot card reading. But it's unfortunately some of the states absorb those tests for the food industry, and they don't want to get off them out of for historical purposes.
Food industry, because their levels of mold and these types of things are
way higher.
A lot, way higher. They've really put this lid on us, and with a very tight restrictions. Yet all these other crops out there are, so many times higher are allowed for it to go, out to sale.
Yeah. It's we always joke around. It's like we're regulated like it's uranium, but
we are. It's crazy how regulated our industry is, and especially
it is nuts. And I don't know how it coexists when there's zero regulation on the hemp side of it. I mean, I don't know what the answer is, somewhere in between, but like those two regulatory paradigms can't coexist. They're very, unfortunately, cannibalistic to one another.
Yeah, we do have a panel at CannMed that's going to deal with accreditation, PTs, because there's challenges with how tests are accredited. There is no standard PT that truly represents the cannabis plant. It's really more of a hemp tea, just because they can't ship interstate commerce.
Even that standards company, I think, backed out. Didn't NCI stop? Didn't they shut their doors?
I don't know. I have to look that up.
I thought the Emerald test shut down recently.
Oh, the Emerald test might have shut down. That's right.
Yeah, so that leads us again back to making our own standards, which it's going to lead to more lab to lab discordances and confusion in the field, unfortunately. But
all right. So as we're winding down here, I got some rapid fire questions for you. We got 4 of them. What's the most underrated cannabinoid right now?
Well, Dustin Sulak just gave a great presentation on CBG, but maybe that's kind of yesteryear, He's presenting on that at CanMed. I think he just gave a talk on the Coffee Talk thing. But I'm very interested in the Barons, just because we have this huge Ozempic craze going on, right?
Yes.
And what about THCV? There was this recent paper showing like a 10% loss in body weight or something for after 90 days on THCV. I'm like, that's a pretty remarkable study, considering the side effect profile of THCV probably doesn't include blindness and a lot of these other things that are being reported with Ozempic.
Yeah.
So that seems to be that could be a big pharmaceutical hit.
Yeah, because I imagine THCV might be a replacement for Ozempic or even Adderall.
Really? Why Adderall?
You look at some of the data that's been coming out. Well, it's if you look at some of the data behind, at least what I've seen, and actually I think someone's presenting on THCV at CanMed. I'll have to go back and look. I thought I saw an abstract about this, but it has potential for everything from weight management to blood sugar control, mental clarity. So they're seeing some early signs that it may be a better alternative than what's on the market today from the traditional pharmaceutical industry.
Mental clarity. That's the first time I heard that with THCV, so that's interesting. Very, very interesting.
Now, granted, I'm not an expert in that field, but we do see a lot of abstracts that come to CanMed, so it just gives us a flavor of who's working on what and where the industry might be going.
Yeah.
I like the canflavins as well, not cannabinoids, but those are, again, compounds that the plant doesn't make in very high concentrations, so that there's maybe a synthesis argument there. And then kynurenic acid is one that has shown up in the literature recently, which is in the leaves. Okay. That's a tryptamine. So we've got to keep our eyes on
that's a psychedelic.
I think on that, because that's something that maybe the entire juicing side of the medicinal aspects here is forgetting the fact that, hey, there's different compounds in the leaves than are in the flowers. And
interesting.
Oh. Yeah.
So it's THCV in a lot of plants is at a very low number. Have you heard of any plants out there that have high THCV? Because this is a very interesting cannabinoid for sure.
I've only seen ones that are like 7 to 10%, and it's very rarely ever more than the THC. So they always have to like fractionally distill it or pull it out from THC. I don't know yet of a plant that does a 20-to-1 ratio, if you will, like we see with CBD or THC.
All right.
But that might be a question for the folks down. I think Allison Holloway might be presenting on that topic. Down at CanMed. I know they've been doing a lot of work on THCV, and I don't know if Ryan Lynch is going. I think Todd Michael's going. He did some mapping of the THCV genes in his recent Nature paper, so they may have a better handle on it. There's a second gene that they think's involved, and maybe they're finding ways to breed it to higher levels than the 5-carbon chain that's in there.
What's the biggest mistake you see growers making time and time again in our industry? Just what's the biggest mistake you see our industry making over and over again?
Oh, that's a good question.
Of course, I imagine, Mike, you would probably have a better perspective on this.
My first thing would be is the wow. One is not understanding that that fertilizers do make a difference for sure, But,
Oh yeah, that in the substrate, like.
Oh, let's talk about the microbiome of substrates, because
Oh, God, yeah.
there's a panel on that. And
Yeah, yeah,
don't give away
that's
too
an
much
area
of the
where-
juice, but the microbiomes and plants, and how's that affecting the growth of the plant and the health of the plant?
I think we only recently began to like recognize that, because it should have struck us many years ago. Like Digipath and Cindy Orsner at one time sent us some people's cocoa. It's had to be 2017 or something. And we sequenced it, and we were shocked to find that there was a microbiome in there. I think in that case it was Fusarium. That was the source of what was screwing up their growth.
Yeah.
And that was just kind of rattling around in my head for years. And then this year, finally, Zamir dug his head into that and said, no, we're going to go through and sequence all the different substrates I work with. And we're going to sequence the roots, we're going to sequence the stems, we're going to sequence the flowers, we're going to sequence the leaves, we're going to sequence everything.
Yeah.
And he's got really interesting data showing that, yeah, the microbiome that's in your substrate can certainly get into your plant. And there are ways of cleaning plants, obviously, with some tissue culture. But I think one of the things he found was that when you tissue culture clean a plant, if you don't reintroduce a diverse microbiome after that, it becomes kind of like blowing a bubble. And then the first thing that gets in there takes it all, right? It's like a vacant niche. And so if you're really exposed to Fusarium coming back, if you don't reintroduce a diverse microbiome on top of it after you've cleaned it, you just can't keep the plant sterile forever. And the sterilization process is important to get rid of the pathogens you don't want. But when you reintroduce this, your substrate is going to feed it. And if your substrate is dirty, it's going to come in through there, and you're going to be back to ground zero. Yeah. So I think that's an overlooked area, as you brought up, is the nutrients and the whole substrate. A lot of people are assuming those things are sterile or don't have a microbiome, but they in fact play a big role in colonizing the plant.
Yeah, we have tablets we call our voodoo tabs, and we look at the plant holistically, and we've always looked at the microbiome, and we realize that it makes a difference. I realized that when I was in my traditional years, when I was growing outdoors, I would find a certain type of a soil, and we call it super soil. And if we found these areas with a super soil, and those plants grew like unbelievable, like they were on steroids. And it was because of the microbiome that was in that soil. So that was the first time I saw that.
I used some of those voodoo tablets you had actually last year in regard. Yes, and I think we even sequenced those. I got to dig it up.
Oh, like good. I'd love to see that. Oh, yeah.
Yeah.
They work really well. Oh,
I don't think I did a proper study like before and after. I think I just used them and said, "Hey, let's sequence it." And so I don't have a great control, but at least I might be able to see that the microbes that you've got listed on the packet I can find in the stem, later. But I think that's particularly important in if you don't have beneficial microbes present, your plant's going to get overcolonized with the wrong things. Yeah. And that's important in states that have these. You got to be careful in the states of these total tests. This is one reason we've been trying to ask regulators to like loosen up on these total tests, because they're basically a de facto ban on beneficial microbes. And if you can't have, because the beneficial microbes will trigger these total tests, right? So if you can't have beneficial microbes, then what are you left with? You're left with like playing the cat and mouse game on the pesticides, and mixing 3 or 4 low concentration pesticides so that you don't break the limits on those. And that's a horrible game that I don't think we'll ever win, because then the list of pesticides has to grow to like thousands, right? They have 66 in some states, but there's a nice Canadian study out there that looked at 327 pesticides, and they can find off-list pesticides getting used when you ban the 66, right? Yep. So that cat and mouse game, I'd rather play that with microbes, because there's plenty of beneficial microbes out there you can use to colonize the plant and push out the pathogenic ones.
Exactly. Yeah. There's more good guys than bad guys, and the bad guys can't take hold. So.
But in many states, those are sort of penalized, but based on the total testing. So it's very important, I think, for growers. Obviously, they know this and preaching to the choir, but depending on the state you're in, the way you grow has got to change based on the regulations.
Yeah, that's true.
Unfortunately.
So what's the biggest myth in cannabis today? Cannabis
biggest
science
myth in
today.
cannabis?
Biggest myth in cannabis science today. Yeah.
I think it's the cannabis schizophrenia link. I think that is bogus.
Really? I've never believed that. You know what? I got into a debate with a woman once in London about that. She brought that up, and I just, You know, I know because when we used to be caretakers for patients up in Canada, there was one gentleman that we did growing for, and he said that his son smokes cannabis because it controls the voices that he hears in his head. He was schizophrenic, and it calmed him down.
That is the argument Ziva Cooper has always raised, who's done great work in this area, which is that there are many studies that show better mental acuity in schizophrenics that are cannabis users, which begs the question of the direction of the association. Okay, yeah, cannabis use is associated with schizophrenics, but so is a lot of drug use. A lot of it is genetic. They have drug-seeking behavior, and maybe they're seeking cannabis use to appease their symptoms. But we also have this issue with CBD is in trials for treating paranoia and for psychosis, right? So most of these studies that go and try to make this link between cannabis and schizophrenia, they never have chemotyped cannabis floating around. They just look at users on a whole basis that, all right, these guys are users, those aren't users. We don't know what they're using, how they're using it. We don't know if it's got pesticides, and they try to draw these correlations. Yeah. But those correlations, you can't always determine whether it's causative or not. And I think Ziva's work, where she highlights the fact that there's a genetic predisposition to schizophrenia that's really high, and that there is better cognitive performance in schizophrenics or cannabis users in some studies, that implies the directionality of this might be maybe they're seeking out CBD, and they don't recognize that the plants are really low in that. And yeah, THC, I do see the literature. THC can drive paranoia. I get that, but does it permanently trigger it in people? I think is a harder question to answer. What drives me nuts about all the studies is that all of these pesticides that we're trying to screen out of the cannabis field, they're all neurotoxins. I would be biologically, I would be more concerned about myclobutanol, which forms hydrogen cyanide after you smoke it, causing schizophrenia, than THC. Yet we don't have a reading on how much of that is out there. We know prior to California putting that on the testing list, SC Labs was finding that like 40% of the pesticide fails were myclobutanol. Yeah. It was Eagle 20 was just used rampantly. So if you extract that into a pen, it's in high concentration and it ends up in some of these patients, then we're going to get really confusing signals on what's causing schizophrenia. And that one I feel is a major public health scare that's being used when their attention should be on cannabinoid hyperemesis, which is a real quantifiable diagnosis that we have much harder data on. And then the genetics of it makes sense, actually. When you look at what Ethan found, you're like, okay, you give those people synthetic cannabinoids that are 10x or 20x more potent, and you can trigger sick and vomiting syndrome. You've got a model there that's or cannabinoid hyperemesis. You can CHS. You can trigger it with the synthetics. So there's a much better, I think, argument for that being a cannabis risk that we need to pay attention to than the whole schizophrenia talk.
Okay. If you could snap your fingers and change one thing in the cannabis industry, what would it be? Final question.
Without a question, I would want similar regulations between hemp and cannabis so the two industries aren't fighting each other.
That makes a lot of sense in bringing regulation and do it the right way. And what I never like seeing are the hemp companies lobbying to keep cannabis illegal like they were in Florida. You're like, this has just gone overboard, right? You're now it's a circular firing squad at that point.
Yeah, because that's fundamentally. I mean, it solves the problem that we have with the FDA that it's really not a Schedule I drug if you have the hemp loophole. That's a definition.
Very good point. Very good point.
Yeah, and I didn't want it there either. I definitely want it to be more legal and liberalized everywhere. I just don't think it's ever going to work out well for people if we have. That's the schizophrenia we have, is that there's banking for only the industry that does safety testing, or there is no banking. It's just safety testing, and there is banking for the group that doesn't. I mean, that's just going to create that's going to create incentives that are a train wreck.
Hopefully,
it's sort of the definition of a major policy failure from the U.S. officials.
Yeah. All right. Wow. It has. First of all, how do people get hold of Medicinal Genomics? How do they get hold of you? Because this is important. Every cultivator out there should be using your services and talking to you, because you guys are a wealth of knowledge.
Website's probably the best place, medicinalgenomics.com, and you can reach either Brendan or I at our first last name at medicinalgenomics.com.
All right.
So
very good.
Kevin.McKernan or Brendan.McKernan.
And let's talk about CannMed. That, again, is June 17th to the 20th in Puerto Rico. People want to get tickets. How do they do it?
CanMedEvents.com is the easiest way. Or once again, you could email one of us and we'll connect you to the right people.
CannMedEvents.com. All right. That sounds good. Both Kevin, Brendan, thank you today. It was very informative, a very high level. And if people want to get more of that and really want to get into the weeds and understand cannabis at a scientific level and get some great information and great help and networking, definitely be at CannMed this June 17th in Puerto Rico. Thank you, gentlemen.
All right.
Thanks, Mike. Yeah, we'll see you
Mike.
in a couple of weeks.
Hope to see you guys down there.
You will.